Hello everyone, welcome to the podcast collaboration between Gray Matters Journal and CoNECT, otherwise known as a community of neuroethics, computation, and technology. Before we get started, I just want to talk about a little bit about why we sort of have this collaborative approach. Podcasting is a fantastic way to create conversations about interesting research in neuro-related frontiers, such as engineering, computer science, biology, physics, chemistry. The list really just goes on. It allows people to interact with a spectrum of perspectives to develop a greater understanding of these topics. This collaboration hopes to spur interest in neuroengineering and provide new avenues for those in the community to get involved in work that is fundamentally changing the way we live. We produce interviews with groundbreaking researchers, postdoctorals, and graduate students. And as we interview these experts, we take the great care to really delve deep into the nuances of the work they do and connect it to our own lives. With that being said, we hope you enjoy this conversation and learn more about the amazing work that these pioneering researchers are producing.
All right, and thank you, Abel, for the introduction. My name is Anthony. I'm your other host. And today we welcome Dr. Brunton. She's a professor of biology in the Richard Joan Coleman University Chair at the University of Washington, Seattle. She's also affiliated with the eScience Institute for Data Science, the Paul G. Allen School of Computer Science and Engineering, and the Department of Applied Mathematics. Dr. Brunton's research sits at the intersection of biology, neuroengineering, and data science with a focus on developing data-driven analytic methods to uncover patterns in high-dimensional neurodata. Thank you for joining us here today, Dr. Brunton.
Happy to be here, Anthony and Abel.
And yes, before we dive deep into the research side, could you tell us a little more about your background, your journey, and how your interest got you where you are today?
It's a great question you asked because, I think you gave a really great introduction. It's really very kind of you to say it that way. I think the simpler way to say it is that I like math and I like brains. That's really how I got here. When I was an undergraduate, I was doing research in molecular biophysics, working mostly in microbiology. And then I took a class. I always liked physics and math and was really into that, but I was doing research in biology. And I realized towards the end of this undergraduate, when I was an undergraduate, I realized I really loved a type of math known as dynamical systems control. So using differential equations to model how things change in time. And it occurred to me that what is biology except the spectacular ability of living things to change in time? So I really liked that type of math. And so that actually drew me eventually to neuroscience because this is a type of mathematics that people use a lot to describe neurodynamics and behavior and how nervous systems give rise to the richness and complexity of actual natural behavior. So that's the short version of why I'm here is because I followed the math and now we're doing neuroscience.
Yeah, so how did you sort of how did you merge your mathematical interests into, for example, microbiology?
Great question. So we did actually, my undergraduate research, we were doing structural biology, so there was a bit of modeling that's associated with that, but mostly biophysical modeling. It wasn't a lot of dynamical systems. It was more like just physics, right? So like trying to figure out what are the forces and structures and morphologies of these cells, and that was really interesting to look at. I was also working on electron microscopy, and so there's quite a bit of very quantitative image analysis involved in doing microscopy of all kinds as well. But there wasn't, like I said, a lot of at least the work I was doing, it wasn't a lot of dynamics, and I wanted to look at how things change in time. And that's what we do a lot of in computational systems neuroscience, and that's what drew me to this field.
Yes, and I know that most people kind of figure out their way and their path and careers kind of like an undergraduate. Were you always interested in during high school, or was it undergraduate?
Yeah, so I'm a pretty giant nerd. I've always gone to nerd schools, and so when I was in high school, I was a part of a science, math, and computer science high school program. This was grew up in the East Coast, so this is in Maryland, and as a part of that, I took a bunch of science classes, but also a lot of math and computer science, and knew that I liked that type of very quantitative approach to science. And it was also at that point, I was very fortunate that I actually got to do research at the National Institute of Health for 2 summers when I was in high school. Again, because of the privilege of just kind of being local, the lab was right a mile away from where I lived. So I hung out there for a little while and got to know a lot of the biologists that worked at the NIH at the time. And so through that experience, I knew I liked biology because I liked the kind of questions that they were asking. But I also knew I liked math and engineering and computer science. And so it took me longer to figure out how I can do all of that at the same time. But I've always been interested in biology and quantitative stuff. I've done that all along.
I can really relate to that, honestly.
Good.
That's why I'm kind of sort of finding how to merge my also interest. And that's kind of funny because that's what really drew me to research, right?
Yeah.
You sort of took this biology, physics, all of them, kind of merging them together,
Mm-hmm.
kind of finding this specialty, which is great. Yeah, I'm still finding mine. But yeah.
Me too.
We all find our own paths.
Yeah, we're all finding our own path, kinda.
Yeah.
On the way.
And you get to change your mind.
Yeah, that's
True.
true.
Like the kind of stuff I do now is pretty different than what I did when I was a PhD student. And so I think and I wouldn't be surprised if in 10 years I'm doing something even different still. So.
Right.
Yeah.
It is dynamic. Yeah,
Mhm.
for sure. Yeah.
And just one quick question. And besides research and teaching and doing school stuff, what do you like to do outside of work?
Outside of work?
Yeah. Just any hobbies you have?
Oh my gosh. So I have a couple of work adjacent hobbies because again, I'm a scientist, I can't turn it off. So I've always done art, and art of different kinds, mostly painting and drawing of different kinds. And I've also, I've done fiber arts of various kinds for a long time, like knitting, and I recently got into weaving as well. And so one of the things that I started doing a little more recently is I got into watercolor painting, and mostly because it's portable. It's the easiest type of painting that's really easy to clean up afterwards. Because I have kids at home and we didn't have a lot of space, and so I didn't have a big studio where you can have oil paintings drying for 3 months in a corner, and because someone's gonna touch it, right? So I couldn't have that. So I picked watercolor mostly out of convenience. In any case, so I started watercolor painting for a while, and it became obvious to me that the world does not need me to paint more landscapes. I do paint landscapes, but we do not need one more person to paint watercolor landscapes. But so what I started doing was I started painting insects and other animals, including the ones that a lot of my collaborators are working on. And so I would ask them, hey, you're working on interesting critters. Do you have any charismatic pictures of your interesting critters that you're working on? And they would send me stuff, and I would paint them. And through that process of painting these animals, I really think I gained a greater appreciation of their behavior, of their morphology, of all the different things that are really special about biology. So for example, I have a friend, my collaborator and friend John Tuttle, who works on proprioception and mechanosensors on legs, right? So for a while I was painting insects and painting every single hair on their legs, and then just being wow, this is just so cool, right? Look at all these hairs and exactly where every single one of them are. This means something. We don't know what it is, but somebody should find out, right?
It's with the patterns.
Yeah, exactly, all the patterns thereof, and just really looking at where they are in their natural habitat, when they're not in a box in a lab, when they're out feeding on a flower or something. Just thinking about more of that naturalist perspective on biology, rather than this laboratory centric view of what it means to study an animal. And we think we need both. I'm not saying that we should go back to just hiking as the only way of doing science like people used to do. Obviously it's both, but I think it is, for me at least, there was a while in the middle I was studying biology from a very molecular cellular perspective, and I think I lost track of what makes life special. And what makes life special is all the things that we actually do in real life, not the things that we can do in a laboratory, but what we do walking around, right? And just not losing track of the awesomeness of nature, right? But in the original sense of the word awesome, full of awe, not the way we use it now. It's everything's awesome. My latte is awesome. Not that kind of awesome, but yeah, it's not awesome with a capital A. It's you go out and you go hiking, you see the Grand Canyon, you see a hawk. Awesome. This is just awesome, right? And that feeling of this is why we do what we do. This is why we're scientists, right? Is because we just we can't help ourselves. We can't turn it off. We're going everywhere and looking at nature and feeling this is just the coolest thing ever. And I get to study a piece of it, and that's really special.
Maybe a better word is awe-inspiring. I don't know.
There too. Yeah, I definitely I like that a lot, Abel. It's I feel inspired. Sometimes if I'm getting a bit mired down in the minutia of doing work of various kinds, I just take a walk, Right, I find it inspiring to be in nature around living things, because we're biologists, right? This is what we do.
And I feel with school, some undergraduates find it hard to balance everything. Mm-hmm. But I feel the most undergraduates that pull through in the end, it's they're doing what they actually love instead
Yes.
of like what other people want them to do.
That's right.
And like, yeah, you love biology, you love physics, you love math, you pull through with everything, and then at the end, like, look at
Try and stop me.
Yeah. You can't really stop that.
Yeah. I guess the other thing that you asked about hobbies and things that I do outside of work. I guess one other hobby that I've gotten into more recently is so I like again my training because of where I gone to school and the courses that I happen to take. I'm actually like I took a lot more quantitative classes that what I would consider like classic organismal biology. And so, for example, I now a bunch of my lab, you know this, Abel, like we've been working a lot on biomechanics. I have never actually taken a class in biomechanics. So I don't know. I mean, I've like been learning it by myself on the side, right? But I've never taken a proper class in the subject. And so I've been doing yoga. And through yoga, going back and looking at the neuroanatomy and anatomy of people, I don't know, like there's all of these books about anatomy and PT and all of this stuff, and like people like there's just like so much knowledge that I never really came across in my own coursework and in my training. I'm learning it backwards with the lens of yoga. So like I just recently got a coloring book. Yeah, I have a yoga anatomy coloring book. Do you guys have these coloring? You know these coloring books I'm talking about, right? Like there's a cell biology one, there's a brain anatomy one. Like I got one about like some like coloring the muscles and the tendons. Like oh, cool attachment points.
That was honestly me when I studied biology. Like instead of like actually drawing like all like the muscles and stuff, like I just fill out colorings.
It's great.
Yeah,
it's great. So I have a pet project right now. Again, Abel, you know this. We've been working on fly neuroanatomy, right? So we've been putting muscles in these like realistic physics engines, and then looking at biomechanically realistic models of laboratory animals like flies and mice and rodents. And so I want to make like an animal neuroanatomy coloring. Maybe not a book, maybe like a page or two. Yeah. You know, I think we have like. It'd be cool, right? Like, really cool. It'd be really cool. I think I've talked a few people in my lab into it, so maybe we'll actually do it one day.
Yeah. Well, that was actually I remember seeing like it was actually recently you posted like this like herding technique. Like,
oh.
Can you tell us a little bit more about that?
Dude. So I saw this on social media the other day. And it's this video that I'll just briefly describe to you. So you know how like they're herding dogs that will herd sheep. You've seen videos of this, drone videos, and it's like crazy what they do, right? They can just hundreds of sheep. They can get them to all go back home and go into a pen, right? So I saw this video, and it was these two border collies, and there was a bunch of ducks in the middle, and half of them were black ducks and half of them were white ducks, and the two border collie. You can tell that they're collaborating with each other through very small movements when they're just like shuffling around and like staring at the ducks in certain ways. They got the ducks sorted into all white ducks on one side and all black ducks on the other side. This took many minutes.
That sounds cool.
What's the neuroanatomy behind that?
Yeah, exactly. What is the decision-making process? Like talk about accumulation of evidence in decision-making, right? And like policy. I mean, I really want to model this as a reinforcement learning problem. I've been talking to it's funny you ask, because like I just talked to one of my postdocs about it earlier today. I was like, what can we do to model this? And like I have some ideas. I think we can do it. It would just be really cool. And so I'm constantly even, not just this is not a walk into nature kind of inspiration, but like obviously I'm like strolling on social media going like, ooh, that's cool. We can work on that. Like this would be a really cool little project. It could even be an undergraduate project,
Exactly. Yeah.
Yep.
Your work sounds really complicated, but it sounds really amazing.
Thanks.
Science.
It's science.
Science.
It's science.
Science is complicated.
Science is. I mean, it's. Yeah. There's a lot. There's a lot. And we go back and forth. And I think one of the things that I really appreciate about my role here at the University of Washington as a professor, as opposed to, for example, being at a research only institution that does not have students and definitely doesn't have undergraduate students, is that we are constantly here immersed in the energy of you guys, right? We have all of your energy as well. And then part of my job is, of course, teaching undergraduate classes. And so I've learned so much from that experience because, you talk to your colleagues, you talk to your graduate students, you talk to your postdocs, and like we're sort of in a little intellectual bubble because we all already speak the same language, right? We skip a bunch of words when we're talking. We kind of know what each other are talking about. And so it's only through pulling way back and teaching an undergraduate class, taking somebody where they're smart, they're interested, they want to know more, but they just don't have experience yet in neuroscience. Okay, how do I explain it to them? Right? What do they need to know?
That's honestly me.
Well, but that's not just you, it's everyone, right? But it's really good for me to have to do that all the time. right?
It's like a constant like reminder, you reflecting back to like the old knowledge and stuff like that. So it's
Yeah,
really good.
and I think it's so great that we get to not only produce new knowledge but share that knowledge
Yes.
with people who want to know.
Yeah.
Exactly. I like talking to my kids for the same reason, right? They're like they kind of use me like Wikipedia sometimes, but that's okay.
Ask about anything.
That's all right.
Yeah. No, teaching is really great. Yeah. Do you want to delve into the research?
Yeah. So, by the way, I just want to say your art is really cool. I did take a look at the websites, and if you want to check it out, it's under Dr. Brunton's for UW, University of Washington. And so, like, moving into a specific topic, while I was looking at your article, and your lab focuses on like a lot of various projects, and I had gone to take a chance to look at them and a few of your publications, and I found one thing that interested me was the multi-model deep learning for natural human neural recordings. You your paper like talked about how you can produce, you can predict about like natural arm movements in the future, and basically with this paper, like what got you into studying this, and like could you explain more about that paper and like the data in it? Yeah,
it's a very interesting project that you brought up, and that's a project that I will it goes back to my first graduate student that I mentored, Nancy Wong, who was a graduate student here at University of Washington in computer science and engineering. And so she and I started around the same time. Like she was a brand new graduate student, and I was a brand new assistant professor. And we randomly met actually at a neuroengineering event that was hosted by they had a different acronym back then. It wasn't the CNT, it was a CSNE or something like that. Anyway, it was like a mixture event. I met her. We like immediately got along and had a lot of interests in common. And so that project started because of Nancy. That was her project. And what we realized was that at Harborview Hospital in the neurosurgery unit, they were on a regular basis doing these kinds of surgeries for managing folks who have intractable epilepsy. Fortunately now there's a lot of medications that are people with epilepsy are able to use to manage their symptoms, and it does actually work really well for a majority of them. But for a small fraction of them, they've tried all the drugs. It's not really working for them for managing their symptoms. And they have a type of epilepsy called they have a focus, right? There's a specific part of their brain, like one small piece of tissue deep inside their brain, that is usually the origin of their seizures. So they end up in the hospital in Harborview, and they're being cared by one of our collaborators, either Dr. Jeff Hogeman or Dr. Andrew Koh at the time, and they go in for a 7-day procedure. So on a Tuesday they go in, they get a craniotomy, so they open up their brain. They do a durotomy, so they open up the little soft piece of tissue that's sort of like a helmet of your brain. And they have electrodes implanted on their brain surface. And then they sit in the hospital for the next 7 days, being recorded from 24/7, continuous recordings. And there's video and there's audio. Okay? This was like a gold mine of a data set.
Wow.
Because those of you who've done experiments in neurophysiology, and especially human neurophysiology, know how difficult it is to get half an hour's worth of brain recordings. Here we have 24/7 for a whole week.
Wow.
Okay?
Wow.
So when Nancy realized that, she went and talked to them and was like, oh, that's super cool that you have this data set. What are you doing with it? And they said, nothing. We deleted after the patient is released from the hospital. And so we went about, and we did. It was a lot of paperwork. Okay? A lot of paperwork later, we got permission to ask every patient who comes in for this procedure for their informed consent for us to keep their data set after they're released from the hospital. So everything is done for clinical purposes. We don't do anything in addition. The monitoring, the recording, everything is already done anyway. All we ask for is permission to keep their data set instead of deleting it afterwards. And so we started doing this. We did this for many, many years and collected a gigantic data set of many, many, many people, where we have these very long naturalistic recordings. The other difference besides the length of the recordings is that they're not being asked to do anything. Yeah. Right? So in a lot of these experiments, it's at least spontaneous. It's not natural. It's not like they're not outside playing tennis.
Yeah.
Right? They have epilepsy, they're in a hospital. So it's not like entirely natural,
Yeah.
you know? But it's definitely naturalistic, and it's also uninstructed. So, for example, a lot of people study what your brain has to do to, like I'm going to reach out for a cup, like reach for something, right? And usually it's like a graduate student or researcher telling the person, okay, go now reach for the cup. So they do the thing, right? And then do that 100 times. So they do it 100 times. Okay, that's the data set. What we have is a similar motion, but when they're reaching out, they're like reaching out for this coffee cup, not because somebody told them to, but because they wanted coffee, or because they wanted to point at something, or because they wanted to scratch their head.
Yeah.
So this movement they're making with their arms, right, it's very similar to the instructive movement, but they're doing it for naturalistic purposes. Nobody's telling them to do it. They're doing it because they want to. They're doing it when they're tired. They're doing it in the morning. They're doing it in the evening. They're doing it in the middle of the night when they wake up and they couldn't sleep. They're doing it when they're having a conversation with a family member. They're doing it when they're alone and they're like reading a book, right? That's what we had. And then nobody had this data set before. And so that was the genesis of this data collection process. And then after Nancy graduated, there was a variety of different folks who joined our team and also contributed to the effort. And the paper that you were talking about, Anthony, was sort of one of the culminations of that series of papers where we started, curating the data set, making it nicer and nicer, and then developing machine learning models to try to understand what are the correlates, the neural correlates of these very naturalistic movements that the people were making.
Awesome.
And I did see that in your paper. You said.
I also pun, so the puns are always intended.
But yeah, I also see that in the paper you had 670 hours of recording.
Yeah.
Was that to you was a lot? Because when I see it, it's definitely a lot.
It's a lot.
How many patients was that? Or?
So the data set, for that particular paper, we ended up using 12 people.
12 people.
We had more, but there was a variety of reasons that we picked those 12 specific individuals because the data set, like I said, the data set was opportunistic. And so there were certain people where just because of, some of them they didn't move that much, or the electrodes weren't in quite the right place for us to analyze arm movements. They were just in different parts of the brain, a variety of reasons, right? And so we picked those 12 individuals out of a much larger data set because the data was particularly suitable for doing the kind of analysis that we were doing. So 12 people, and we tended to analyze about 4 days each. And I think that ends up being about right for the number that you said.
Yeah.
And not only did we analyze the data set, we released the data set. So I believe very strongly in open science. And part of open science is not only are we publishing papers to release to the public, we also publish the data set. And so we went through a pretty big effort curating and cleaning up and making the data set in such a way that it's actually now on the internet in a very nice container in a way that other people can use it for their research if they wanted to. Obviously, we're not releasing private videos of the individuals, but we have informed consent and got institutional approval to release the tracked positions of the key points. So not the videos, but the XY positions of their movements over time. So we are using computer vision to track hundreds of hours of video, thousands of hours of video. We're tracking the nose, the 2 shoulder joints, the 2 elbows, and then the 2 wrists, and kind of like the orientation of the torso, like if they're slouching or something, we track that. And we release that as part of the data set, in addition to all of the brain data.
Yeah.
That sounds really cool. And in your paper, you also wrote that you were able to predict future movements up to 800 millisecond before movement initiation. And people may think that this is a small number, but I think in order to predict the future movement, that is it's a really cool data.
It is.
Yeah. And so with this neural decoding and brain computer interfaces, you have 4 publications, I think, on your website.
Maybe.
Probably that or more.
I'm sorry.
Are you still planning to do further research within this field? Because you did mention that this was a collaboration with you and...
Yeah, so it's a bunch of people involved. So my most closest collaborator was Rajesh Rao in computer science, and our clinical collaborators at Harborview in the neurosurgery clinic. I also have a different project on human neuroscience with my friend Alec Wedge, who is an MD PhD psychiatrist at University of Minnesota. And that one is not from neurosurgery, but we do have a pretty large data set. And that one, it's a neuropsychiatric project where we're trying to predict psychiatric symptoms and presentations. So different goals, but we're actually using the same model that we developed to predict movements, to see if we can predict people who have suicidal ideation. So that one has a different set of mental health goals involved. So that project is also ongoing. Yeah, go ahead.
Sorry if I interrupted.
No.
Have you heard of like Natasha Kolowat's like research?
I have not.
Okay.
Yeah.
I don't know. I think that that kind of reminded me because I think they were also trying to find some like biomarkers.
There's a lot of that stuff going on, yeah.
With regards to suicide.
Yeah. But I think the bulk of my lab, the research over that we're currently doing, that we're likely to continue doing for the next, let's say, 5 or 10 years, is largely focused on the combination of models of the nervous system with the biomechanics of the body, focus on animal models because that's where the data sets are. So there's a big effort right now in my lab to build these virtual animals that run around in essentially a video game engine. You know how you can like, I don't know, my son was playing Red Dead Redemption or something, you can ride a horse? You know what I'm talking about, right? You're like riding the horse and there's a little animation of the horse like running around. Yeah, not necessarily in VR, but like they're in a physics environment and you can watch it in VR or you can just watch it on a television screen. But there's an engine that runs exactly how the horse runs forward, right? So we're talking about that, but instead of making it, so like if you're a video game developer, all you really care about is that the animation looks good. It doesn't have to be realistic.
Yeah.
So in our tool, it's a piece of software called Mujoko, which is actually, it originally started...
Minecraft, right?
No, Mujoko.
Oh, okay. Not motion.
We can totally
I
do
love
it in
Minecraft.
Minecraft, though. We can totes do this in Minecraft, dude. Okay. So we're doing it in this tool called Mujoko, which is kind of like a video game engine, except it is biophysically and biomechanically realistic. Like, it respects F equals MA. If you push on something, it pushes back, and it does in a realistic way. And it also, so this is like a research tool for doing virtual physics engines. A lot of people in robotics also study it, also use this tool. It was originated by Emma Todorov, who is a faculty member here at University of Washington as well. But it's sort of now it's kind of gone off. It was with DeepMind, and DeepMind got bought by Google, so now it's a Google thing. Anyway, so we, along with our collaborators, are working on a couple of body models that are very biophysically realistic. There's one of the fruit fly, there's one of the rat, and then I'm not totally involved, but there's a couple of people trying to work on one of the mouse as well. I think in the future there might be like a virtual zoo of animals. Like we can have bats and finches and all kinds of things. So the future usually accelerates, which I think is a cool thing, right? If you think about like the early days of the genome project, it was a while. Like first we could sequence like a gene, right? Like in the 70s, if you sequenced a gene, you got a PhD, just one gene, because it was pretty hard, right? And then
Singer. And then there comes Singer.
Yeah, exactly. And then they figured out how to actually do a genome of small animals, and then they figured out how to do a genome of much larger genomes like humans, and now sequencing a new organism, a new species that you find in the ocean somewhere, is like, it's not trivial, but it's very doable, right? You send it off to a lab, you pay money, you get a genome back. So if you think about how long that took, right, we're talking about decades. So not months to years, but decades, right? Over the course of decades, it went all the way from we can barely do one gene to, let's just sequence a new genome, not a problem, right? So that's sort of, I don't know where we are on that curve. Like we're at the point where building one animal model that's realistic of one animal is like, it's a lot of work. We're working on it. It's a lot of work. But it is my hope, optimistically, that as we do one, the second one will be easier. And then the third one will be even easier. And eventually we might figure out how to do this in a more automated way that's scalable. And we might be able to do this for other animals. So if you, again, go back to thinking about the physics engine and the horse you might be riding in a video game, right? Once you build one, though, modifying it becomes really easy. Okay? So like if you had a horse, you can make the horse heavier, trivial. Mm-hmm. You can make the horse a little bit bigger, also trivial. You can make it so that one of its legs is hurt. And you can model how it would try to walk and or run when it has an injury in one leg. Now it gets interesting, right? Now we have a neuroscience question. What is the compensatory mechanisms, both at a biomechanical level, like does it have to get stronger in the other leg, right? Is there more strain on the tendons of the other ankle? And also a neurological adaptation problem. How does the nervous system change its control strategy to compensate for the fact that one of its legs is now lame? Right? Over time, I mean, if you haven't had a sports injury or something, you might be like, okay, I'm limping around, like I hurt my ankle, I'm limping around for a while, right? If it's a permanent injury, like an amputation or something, you actually have to just, your nervous system has to deal with it. It needs to learn, all right, I don't have this part of a leg anymore, let's like actually just walk differently. And there's systematic changes, not only locally at that leg and the other leg, but everywhere in your nervous system compensating for this. right? How does that work?
Yeah.
And
fascinating.
really fascinating and really interesting applications to, I don't know, like people who are compensating after injury and stroke, for example, right? I'd either, including immediately, like I hurt my ankle, how do I get off the basketball court? Intermediate term, I'm recovering for a few months, like let's not hurt myself worse. And then over really long time frames, okay, like for the rest of my life, I'm going to have a bum ankle. What am I going to do about this? Right? Like, so all of those levels of compensation, including what are some therapies that we can use? What's the optimal strategy for understanding the neuroadaptation? I don't know. There's just like a lot of questions that we can use, right? And that does not actually involve building new biomechanical models. We can do that with one, because modifying it in small ways without building a new one from scratch is totally accessible, even if, like, I don't know how to build a bat. I really want to build a bat. I have friends who study bats. I really want to buy it, but we're not there yet. No one's building it. We're not building a bat. Eventually we will have a bat.
What do you think, like, are the current limitations, though, in, like, this work? Like,
Current limitations?
yeah.
Actually taking the physics in a serious way becomes very challenging. And these are canonically challenging problems that are well known in the physical and engineering sciences that don't have to do with biology. So, for example, modeling fluids is difficult. Just ask Boeing, right? Or Formula One race car companies. Like, they spend a ton of money trying to model the fluid structure interactions of something going through a liquid of some kind. And that's difficult in itself before you start worrying about the bat and its wings. Okay, so that's challenging. Mixed media and different kinds of surfaces are challenging. Like, we can do walking on surfaces, like a flat surface. It's like, you know, this carpeted ground here, or a wooden ground, or whatever, anything flat, totally cool. If you make it bumpy, we can still do it. If you put mud on it, mud is really hard. Yeah, mud is really hard.
Viscosity, right?
Yeah, the viscosity, and then there's the interface. Like, the moment you go from, you pick up your foot, and then you start stepping into the mud, right? And it's squishy. And at some point you hit the bottom, and then you try to pick up your foot, and it's like sucking you back down a little bit. Like, that's really hard.
Right.
So that's a limitation. The other limitation, I think, is development. Because we're not large language models. We don't go poof into your adult body, right? We have to actually grow into that body. And our nervous system has to be working the entire time. And so I think a lot of the really interesting questions after we handle one static body morphology is, how do you grow into that body shape, and how do you handle aging as that body starts to degrade as well, right? So there's a lot of rich questions there that are going to be really challenging. We're not just doing the biomechanics, we're also doing the nervous system. So this is, again, a really exciting time to be working on this right now because of you've heard of the CoNECT data sets that have been coming out in recent years. Again, this is like in analogy with a genome. It's like the early days of the genome where we got we got one genome, so excited. Next year we might have two. Ooh, like we're at that stage right now. And so I'm both really excited about it, but also like just kind of waiting for more resources to become available. And I feel like it's quite doable. It's coming. Our friends are working on it. But it's a really exciting time, right? Because it's not just the ability to characterize the connectivity of the neurons in your nervous system, but to do it in a comprehensive way. To me, in analogy with the genome, that's the difference to the genome, right? If you only have one gene or two genes or even 100 genes, you make a little network. It's like, okay, this transcription factor activates this one, which turns on this metabolic pathway. Everything's all good, right? Anything you can't explain, you're like, maybe it's a gene that we don't have. We don't know because it's out there somewhere, right? And that's where it's exactly where we were in neuroscience too. We study some brain areas. We have some recordings. We have some data. We're like, okay, here's how it works. Oh, it doesn't work? Maybe it's some other brain area that we don't have yet. You can always point somewhere else, right? So the magic of the CoNECT data sets we're starting to be able to get to is that there's nowhere else to point. Okay, we have the whole thing.
Yeah.
This is it, right? It is either here or somewhere in there. Those are the choices. You can no longer say it's somewhere else.
Okay, got it.
Right? So the comprehensiveness of these data sets and their ability to model them in a really, really comprehensive way, and to put it together with a body that it actually controls, that's where I think the really exciting stuff is. Full disclosure, lots of things we don't know. I'm being a little bombastic. We don't really know much about electrical gap junctions. We don't know much about neuromodulation. And so even within the comprehensive connectome, there's like things that we don't have. So, but nevertheless, we can't, it's like mapping something, right? You map the whole thing. You at least know the lay of the land,
Right.
okay? There's lots of stuff in there we don't know. We don't know about channel conductances. We don't know about neuromodulation. Lots of things about plasticity we don't know. Like lots and lots of things we don't know. But we have the entire map.
Yeah.
Okay, this is it. Here's a good start. And to me, that's really exciting.
Nice.
It's okay when you build a puzzle, the first thing you build is a frame.
Yes.
Yes.
Great analogy. I really like that. It's a place to start, right?
Yeah.
Like
It's a
whatever is going on, it's in here.
It's okay.
I still can't find it. I don't
It's
know where
just
it goes, but I know it's in there.
Yes.
Yep.
Great.
Mm-hmm. Great analogy. I like that, Anthony.
Yeah.
Thank you for that.
Thank you. Yeah, and we're a little bit short on time, but we really appreciate having Dr. Brunton, and we hope you guys took away something, or many things.
I definitely did a lot because my major is biochemistry, and this is like the first time I really got to like dive deep into like a topic within neuroscience. It was really cool to see that.
It was really fun talking to you, and you came up with some really great analogies for our work.
Yeah. All right, thank you.
All right, thank you so
Thank
much for that.
you.